On behalf of the Lamb Foundation, it is my pleasure to welcome you all to the second educational session in our Lambposium in Your Living Room series. My name is Charlene Dunn, and I am the Patient Outreach Coordinator for the Foundation. Thank you for joining us as we bring Lambposium into your living rooms. It really, again, just like last month, it's so exciting to see so many faces and names from across the country and from around the globe. For those of you unable to attend last month's session, just a quick little backstory on this series. When we made the decision to cancel Lambposium, while we didn't know exactly how the next few months would unfold, we did know that we wanted to somehow carry forward the inspiring planning and programming that we had originally scheduled to offer everyone in Cincinnati. With the very enthusiastic support of our already scheduled speakers, we were able to quickly transition our Lambposium sessions to this virtual format, Zoom, that we've all come to know quite well at this point. So again, thank you all for joining us and celebrating this gathering. Before we get started, I would like to take a quick moment to thank the National Disease Research Interchange, or as we know, NDRI, for their ongoing support in our research efforts. The Lamb Foundation partners with NDRI to collect patient-donated surgical tissue samples. And NDRI then works to ensure that these samples are prepared precisely according to the specific needs of our investigators' protocols. And of course, this tissue donation process all begins with our patients, and we're going to learn much more about that this evening. I'd like to cover just a couple of really quick housekeeping items before I turn the evening over to our moderator and our speakers. Microphones will be muted throughout the presentation. When we move to Q&A, we'll ask that you write your question in the chat box, and our moderator will then read the questions to our speakers. Our speakers have agreed to extend our Q&A until 8:15 this evening, which is fantastic. They're very excited and eager to engage with everyone, so don't be shy about asking questions. We'll type them in the chat box and keep Jessica busy as she reads those questions to our speakers. And then our patient and family group discussion will then run from 8:15 till 9 p.m. And with that, it is my sincere pleasure to introduce Jessica Barcelona, who is our moderator for this evening. Jessica is a Lamb liaison in Region 3, which encompasses New York City. Now, she has an interesting health history, but I had to shorten this a little bit tonight. So I just want to say a couple of things. After a few rounds of misdiagnoses, Jessica was eventually diagnosed with Lamb in 2015. And 3 years later, she joined, can we back up that slide, Julie? There we go, thank you. And Jessica was diagnosed with Lamb in 2015. And then 3 years later, she joined our liaison network about the time of our 2018 Lambposium, which was held in Cincinnati. When I introduce our liaison moderators, I really like to share just a little something about their lives outside of Lamb. So for Jessica, I'd like to start with her job title. And I actually need a deep breath for this one. So Jessica's job title is Emergency Field Operations Planning and Training Director of New York City Department of Health, Office of Emergency Preparedness and Response. It's a mouthful. And given the current pandemic, I think it's fair to say that Jessica has really had her hands full in recent months. In addition to this, she also teaches a master's level course in emergency preparedness. Now, what I want you to know is Jessica elaborated on her work and what it entails after she agreed to host this webinar. When I thanked her for saying yes to me, first I quickly corrected myself and said, well, what I mean is thank you for saying yes to the foundation. Jessica's response to me, well, it's a yes to all of us. And I couldn't agree more. Jessica, thank you for hosting this evening, and I'll turn it over to you here.
All right, thank you, Charlene. I'm really excited for tonight's presentation and to be the moderator. And also it's the kickoff to Worldwide Lamb Awareness Month. I am wearing my lovely t-shirt that you probably can't see that came in the mail this morning, so that's fantastic. And with us this evening are Doctors Stephen Hammes and Catherine Wickenheiser-Brokamp, who will be discussing the advancements in Lamb research and how tissue donation from Lamb patients has powered many of these advancements. I'm really eager to hear what they have to share with us. as I'm sure all of you are as well. Before we get started, I want to focus on one of our LAM patients, patient spotlight, on a patient whose lung tissue contributed to the research being conducted by our scientists. So this month, we're spotlighting Lisa Catalano. Lisa is a lung transplant recipient that lives in Punxsutawney, Pennsylvania, near Pittsburgh. She was diagnosed with LAM in May of 2018. After 6 days at a local hospital, she knew something was not right. She traveled to Pittsburgh to an emergency room there, and upon arrival, her oxygen saturation was at 80%. During her week-long stay in the hospital in Pittsburgh, she received the diagnosis of LAM. The doctor asked if she could bring residents into Lisa's room because students may never see another LAM patient in their careers. Lisa said yes, contributing to LAM awareness right away. Lisa was given 2 options by the doctor: be on 15 liters of oxygen for the rest of her life or go on the transplant list. Lisa told me she had too much living to do and that the transplant option was her choice. She underwent evaluation and was listed for transplant in September. Less than 3 weeks later, she got the call and received the transplant on October 15th, 2018, barely 5 months after diagnosis. Lisa connected with the Circle of Hope transplant support group very quickly and was willing to donate tissue to research, despite having had much time to wrap her head around LAM. The transplant happened so quickly, she hadn't even completed the paperwork for the National Disease Research Interchange, or NDRI, but she was willing to do so from her hospital room as she was being prepped for surgery. Lisa recognizes that so much research needs to be done. She was very happy to be able to donate tissue to support LAM research and to help others. More than 2 years post-transplant, Lisa's doing really well. She enjoys spending time gardening, although when talking with her, to me, it sounded like farming. She's got cantaloupes and watermelons and chickens and a whole bunch of things that me as a city girl doesn't really know how to grow or do anything with except cook and eat. Canning and preserving vegetables from her garden and reading. Some of her current reads are up on the slide. Lisa has a great support system that she's really grateful for with her family, including her 85-year-old father and her church community. She gives good hugs, so she says one day, hopefully soon, she'll be able to give those hugs in person once again. Lisa encourages everyone to not give up. We all have a lot of living left to do. And I believe Lisa is with us tonight on the webinar and on the chat, so hopefully she'll be able to join us a little later in our Q&A and discussion. And I really want to thank Lisa for participating in this patient spotlight. All of the patients who have participated in donating their tissue to research, really, we're so grateful for you and for your contributions, because this is how we make advancements in the research and in the LAM community. Now moving to our guest speakers for tonight. First, we have 2 speakers, which is fantastic. We have Dr. Stephen Hammes, who received his MD and PhD in immunology and microbiology from Duke University, followed by a residency in general medicine and a fellowship in endocrinology at the University of California in San Francisco. In 1999, Dr. Hammes joined the faculty at the University of Texas Southwestern Medical Center in Dallas, where he was a W.W. Caruth Scholar in Biomedical Research. Dr. Hammes moved to the University of Rochester in 2009 to become the Lewis S. Wolk Distinguished Professor of Medicine and Chief of Division of Endocrinology, Diabetes, and Metabolism. Dr. Hammes is interested in steroid production and steroid signaling in female and male reproductive tracts, as well as in steroid-sensitive tumors. He is interested in both the positive and negative effects of androgens in the ovary, with a focus on the pathophysiology and treatment of polycystic ovary syndrome, PCOS. In addition, Dr. Hammes studies how androgens and other signals promote prostate cancer proliferation. Finally, Dr. Hammes is interested in the origins and pathophysiology of lymphangioliomyomatosis. So thank you, Dr. Hammes. We also have with us tonight Dr. Wickenhauser Brokamp, who received her MD and PhD in medicine and development biology at the University of Cincinnati, followed by residency training in pathology and a fellowship in laboratory research at Washington University in St. Louis. In 2003, she returned to Cincinnati, where she developed her independent research program and clinical expertise in pulmonary pathology at the University of Cincinnati and Children's Hospital Medical Center. Her research focuses on understanding the mechanisms driving lung disease. She is also an avid teacher with multiple leadership roles in training physician scientists. Dr. Wickenhauser Brokamp became involved in LAM research shortly after returning to Cincinnati, working closely with both Drs. Frank McCormick and Nishant Gupta to establish safe and effective approaches to LAM diagnosis, conduct research to further understand LAM development and progression, and establish clinical guidelines for LAM diagnosis and management. Dr. Wickenhauser Brokamp is passionate about fully understanding lung disease in patients in the clinic and then using patient-derived tissues to identify novel approaches to diagnose, predict outcomes, and treat patients. In addition to all of this, both our presenters are current members of our Scientific Advisory Board, which advises the Foundation on all scientific matters and formulates the direction of LAM research. You can find their full bios, because there's more, for both of our speakers on the Foundation website. With that, I'll turn it to Dr. Hammes to get us started.
Okay, thank you very much for that introduction. I'm going to pull up my slide. So thank you very much again, Jessica. I really appreciate it. It's great to be here. What I thought I would do was talk a little bit about some of the work that we've done and how it relates to the LAM Foundation and how it relates to lymphangioliomyomatosis. So the original title I had was this kind of long one, using mouse models to develop new knowledge and potential diagnostic and therapeutic targets for LAM. But then as I was sort of thinking about what I'm going to talk about and what Dr. Wickenhauser Brokamp is going to talk about, came up with another title that's a little sort of to the point, which is how to learn more about LAM by going from bench to bedside and back again. Because what we're really going to talk about is how things you discover in the lab can then lead to discoveries on how to treat and diagnose LAM, and then taking tissues from patients and getting information just from seeing patients and diagnosing them and treating them. We can learn things that we can then take back to the lab. And so there's this crosstalk, this interaction that's always going on, and it's absolutely critical. And I think I've never seen a disease where it's more apparent than in LAM. And I think that's in large part due to the LAM Foundation and all the patients and all the doctors and all the scientists who really interact so well together. And I'll keep emphasizing that. We all know that LAM is a cystic lung disease, right? And it leads to, decreased breathing, and it's a slowly progressive disease. But when you look more, and we all know when you look more closely at the cysts, so this is just sort of a slide taken from a lung sample, you see that these cysts are surrounded by these little tumors. And when you zoom in on the tumors, it turns out that these are smooth muscle cell-like tumors. So there are markers, biological markers or biochemical markers you can use to identify a smooth muscle cell. But when you just look at them, features that they have are they have a lot of pink in them. And they're kind of flattened cells. So the dark purple are the nuclei, and you can see how the nuclei are just kind of flattened in these cells. They're heterogeneous tumors, but they do have these smooth muscle cell bases, and I'm going to come back to that in a minute. So the main point is I think everybody knows, is that LAM is a lung disease, but it really is a cancer. It's a tumor. And so understanding how to treat it really is going to be involved in understanding how these tumors grow as well. Okay? So when I think about LAM, and many other people think the same way, there are 4 features that we keep in mind. One is that LAM cells contain inactivating mutations in tuberous sclerosis genes, the TSC genes, TSC1 and TSC2. So both of these work together in a complex to inhibit the protein mTOR. And mTOR is really important for proliferation and migration and metastasis of cells. And so normally, TSC is holding mTOR in check, and there's very little activity. But when you have a mutation, such as in the disease tuberous sclerosis or in LAM, now mTOR is constitutively on, and this leads to the proliferation, and those smooth muscle cell tumors grow. And of course, TOR stands for target of rapamycin. So drugs like rapamycin And analogs like sirolimus, the way they work is they inhibit mTOR, and so then the tumors will grow more slowly and will keep the disease in check. Of course, the problem is that rapamycin and sirolimus, they don't kill LAM cells, they just slow them down. And they can't slow them down completely, and they don't always slow them down forever. Plus, it has its own set of side effects and other things. So it's a great drug, and it's a miracle drug, and it really was discovered and got FDA approval through the work of Frank McCormick and other people in the LAM Foundation. But obviously, we need better treatments, and so we're always on the lookout. So that's the TSC genes. Number 2 is LAM cells express these melanocytic markers. These are proteins that you normally see in melanoma or in melanocytes, which are basically skin cells that give our skin their color, and they get darker in the sunlight, for example. And melanoma, which is when these become cancerous, have even higher expression of these markers. And for some reason, LAM cells express these markers, and we really don't understand why they express it and what controls the markers or what they do, what the biological function they have within the tumors. But it turns out that a lot of cancers, including breast cancer, do express these melanoma markers. And so we're going to come back to them when I talk a little bit about some of the work we've been doing in our laboratory. The third thing, which I think everybody knows, is LAM is found almost exclusively in women. It's very rare that you see it in men. Some men will have very small cysts. Men who have tuberous sclerosis will sometimes have small cysts, but it doesn't really cause clinically relevant disease. And obviously in women it does. And it does appear to be estrogen sensitive because during pregnancy, when people are taking birth control, LAM tends to worsen, and postmenopausally LAM tends to stabilize. So it's only in women, and it appears to be estrogen sensitive. And finally, there's some evidence that LAM cells are metastatic. You can see it in the bloodstream, you can see it in lymph tissue. And so the thought is that it's very likely coming from somewhere else. So the way I got started in this, it really is all about the LAM Foundation. So the first question we asked is, where does LAM come from? And the reason why we even asked that question is because about 10 years ago, Lisa Henske asked me to come give a talk at the Lamposium. So as was mentioned in my introduction, I'm an endocrinologist, really mostly focused on reproductive endocrinology, and my clinical and research interests are focused on estrogen and androgens and how they signal and how they regulate problems with fertility, problems with reproduction, but also how they regulate tumors. So you can see why it was I was a good fit. And so Lisa brought me in really just to talk about how steroids signal and just to interact, to bring an endocrinologist sort of into the fold. And that first Lamposium was really, and all of you who've been and know, it's a life-changing event to spend time interacting with the scientists and the physicians, and especially the patients and their families. It really is amazing. And so I was hooked immediately, and I wanted to be able to do more. And so I think it was the actual evening of the big Lamposium, of the big event, when afterwards we were talking about, where does LAM come from? And the second question we asked is, well, why is LAM primarily in women? And so our thought was, well, it's a smooth muscle cell tumor, it's metastatic, so it's coming from somewhere else, and it seems to be sensitive to estrogen. So what tissue do women have that men don't have that has smooth muscle cells in it? And so the answer we came up with was a uterus. And so the uterus contains a number of different layers, but one of the layers is I boxed it here, is the myometrium. And the myometrium is basically a smooth muscle cell layer that's found in the uterus. And of course, sometimes the smooth muscles grow out of control, and you can get fibroids or leiomyomas. And so the name, lymphangioliomyomatosis, comes from the fact that they look like lyomas. They have that pink staining and those flattened blue nuclei here. And so there always was this thought that there might be some similarity, at least in the way they appear. And we wondered whether perhaps if you have a mutation in TSC1 or TSC2 within the uterus, whether that will become a metastatic tumor that can then go to the lungs. So could LAM be coming from the uterus? So it turns out right about that same time, this was 2011, there was a study out of Japan where they took 10 consecutive LAM patients, and they took out their uteri for various reasons, mostly because they had fibroids. And they looked for microscopic LAM lesions, and 9 out of the 10 patients did have LAM lesions within their uterus. And it turns out that since that time, almost every uterus that's removed from a LAM patient is now studied by somebody within the foundation or somebody within our network, and essentially all of them have LAM lesions within them. So there certainly seems to be some kind of connection, whereas patients who have LAM also have LAM lesions in the uterus. It doesn't prove that one comes from the other, but at least they're both present in most women. So the question we asked, because we like to work with mice was what happens if we specifically knock out the tuberous sclerosis 2 gene, TSC2 gene, only in the uterus of mice. And we can do that using mouse genetics. And the question is, will mice develop LAM tumors in the myometrium? And the second question is, will the mice develop metastatic tumors that go from the myometrium to the lungs? Because we're only removing TSC2 in the uterus. So if we see a tumor in the lungs, it has to have come from the uterus, okay? So I'm not going to show you a lot of data, but I'm going to show you one slide that kind of says it all. So you don't have to be a reproductive endocrinologist or a biologist of any kind to say that the knockout uterus looks really different than the control uterus. Here's a normal uterus. These are basically 2 normal uteri, and here is a knockout uterus. It is huge. And it's full of all these myometrial smooth muscle cells. And here is a giant smooth muscle cell tumor located in this knockout animal. So the funny thing is I actually took these photos on my iPhone. And my wife and I share a camera. We share all of our photos. So my wife found this a day or 2 later, and she was looking at the photos and was really wondering what I'm doing in lab creating these monstrous uteri. But you can see here, they're clearly abnormal. And when you looked at these tumors, they look exactly like LAM tumors. They have all the features of LAM. mTOR is activated. They've got estrogen receptors, progesterone receptors. They express all of those melanocytic markers that I mentioned before, including HMB45, which is the one that we use to diagnose LAM. And so they, for all intents and purposes, we created a mouse model for LAM simply by knocking out TSC2 in the uterus. This just shows you the lungs of an animal. It turns out about 50% of these animals did develop metastases to the lungs. You can see here's a big LAM tumor and little smaller ones. You can zoom in and you can see that pink staining, and they look exactly like the uterine tumors. And in fact, they are exactly the same thing. And if we remove the uterus before metastasis can occur in these knockout animals, none of the animals develop lung lesions. So clearly it's coming from the lungs, or from the uterus to the lungs. Now, you don't get that cystic lung disease that you see in LAM, and we think the reason why is because our tumors are pretty aggressive. And you can see that they're growing really, really quickly. So before you can form the cystic lung disease, it's not a slow-growing tumor, it's a fast-growing tumor. So we think they actually get big before they can form the cystic lung disease. But the point here is that you're able to knock out TSC2 in a uterus, and then they will develop tumors in the lungs, suggesting that perhaps LAM does metastasize from the uterus to the lungs. And the important thing to remember is that these uterine myometrial layers, they are very estrogen sensitive. So now we can take our mouse model and we can get rid of estrogen and we can see what happens to these tumors. So this just shows you, this is an 18-week-old normal mouse. Here's the uterus, and here's a knockout animal. And again, you can see at 18 weeks, this uterus is really big, and the sort of paler color, these are all leiomyomas. These are all smooth muscle cell tumors. What happens if we take these 18-week-old animals and we take out their ovaries? And so they're no longer able to make any estrogen. We're basically putting them into a supermenopause. And you can see if you look at them 10 weeks later, now the knockout uterus looks almost the same as the control or wild type uterus. So if you get rid of estrogen, these tumors not only don't grow, they actually kind of melt away. And the uterus starts looking normal again. So in this model, these tumors are 100% estrogen sensitive. If we treat with a drug called letrozole, which is an aromatase inhibitor, so letrozole will inhibit the production of estradiol, you can see the exact same thing. When you treat with aromatase, they're damaged goods because they were big once, but they're about the same size as the wild type or control uterus. So this really tells you that, again, that you need estrogen in order for these tumors to grow. I should mention that letrozole, this aromatase inhibitor, is commonly used in breast cancer to try and eliminate estrogen production. And it's really a first-line defense for breast cancer tumors that are estrogen receptor positive. So with this background showing that estrogen was really important, we said, okay, let's see if we can figure out what genes within these tumors are upregulated by estrogen. What is estrogen really turning on? And so what we did is we compared the tumors to the controls, and we did something called RNA-seq analysis. And you're going to hear a little bit more about this in the second talk. But basically what we did is we just took a knockout uterus and a wild type uterus or control uterus. We treated both the animals with estrogen for a few days. And then we collected RNA from the uteri, and we just looked to see which RNAs are up and which RNAs are down. Red means up, green means down. And you can see there's a lot of red in the knockout uterus here, whereas the green is in the control uterus. So there's a lot of genes that are turned on. And what surprised us is the genes that were turned on the most were those very same melanocytic markers that I mentioned a few slides back, including that HMB45 target called melana that was profoundly upregulated by estrogen. So we still don't know exactly why these markers are being upregulated, but we do now know why. We do now know what's causing them to be upregulated, and that is estrogen. In the absence of estrogen, these melanocytic markers pretty much disappear. And so we started to look at these melanocytic markers, and there was one that was upregulated the most by estrogen, and it was something called GPNMB. And what this is it's a marker that's highly expressed in melanoma. It's also highly expressed in some kinds of breast cancers, and it's believed to be very tumorigenic. It's expressed at the cell surface of these cancers, and it actually promotes them to grow. And in fact, there are drugs that are being developed that directly bind to the cell surface marker because it's only on the cell surface in cancers. So there are melanoma drugs that are being developed that target GPNMB because it's on the cell surface, and so it's approachable by certain kinds of medications. And as you can see in a uterus from our knockout animal, there's a lot of green fluorescence here. So we're labeling GPNMB, and there's a ton of GPNMB, and there's essentially none in a normal control uterus. But if we take this knockout animal and we get rid of estrogen, same thing, get rid of the ovaries, GPNMB expression goes down to almost zero. Again, proving that estrogen is really important in order for the expression of GPNMB. So then we thought, well, GPNMB has never been reported to be expressed in human lamb, but boy, it's a great marker. It's really easy to see. So we actually took some human samples. This is a lamb lung, and you can see here are some lamb lesions. Here's some normal lung, maybe some more lamb lesions on the side here. And when we stain them with GPNMB, you can see how much they light up. The green staining suggests there's a ton of GPNMB expressed in lamb lesions, and in normal lung, there's very little. And this is a much brighter and more specific staining than even what you see with that HMB45 antibody, which is what we use to diagnose lamb now. So it's a new biomarker that we can use to detect lamb cells. This is a lamb lesion in a uterus. Here's the lamb lesion. Here's the normal uterus. They look a little bit different. It's a little paler, the lamb lesion, compared to the normal myometrium. If we stain for GPNMB, the lamb lesion lights up like a Christmas tree in green here, but the rest of the tumor, the rest of the normal uterus doesn't light up at all. It's just blue. And so it really does suggest that, again, it's a very specific antibody, and it doesn't just recognize any smooth muscle cell, it recognizes lamb lesions. So we're very excited because we made a discovery in a mouse model, and we were able to translate it back to human tissues and to show that what we discovered in the mouse is actually useful in the human tissue as well. So there's one final experiment I'm going to show you. Remember I mentioned that GPNMB is expressed on the cell surface of lamb cells or tumor cells. And it turns out that it gets released very efficiently away from the surface and into the surrounding area. And in fact, we hypothesize that maybe GPNMB will get released into the blood. So instead of having to do a biopsy of a patient's lungs or uterus to find whether GPNMB is expressed, maybe we can just look for GPNMB in the blood of patients who have lamb. So here we're detecting GPNMB in 10 patients who have lamb versus 10 controls. And there is a little bit of overlap, but you can see in general that there's significantly more GPNMB that we can find in lamb blood, and blood from patients with lamb, than in control patients. So it really does suggest that this is a biomarker that we may be able to use, not necessarily to detect lamb, since there is some overlap, but perhaps it's a simple blood test that eventually we'll be able to use to follow lamb as it progresses, right? As the disease progresses, maybe the amount of GPNMB will go up in an individual. So in addition to VEGF-D and other markers, maybe this will be another biomarker we can use. And again, it was discovered in a mouse model and then translated back to human patients. We're also very interested in GPNMB because it has properties that promote cancer. And so we're studying whether we can use it actually as a target to try and slow down lamb progression. So again, an example of how we can work with patients, we can work with physicians, and we can work in the lab to try and make new discoveries that will help us diagnose and treat lamb. And this last slide is just a picture of the University of Rochester. The interesting thing about this project is it would have gone nowhere except when I proposed to do it, I actually got a pilot award from the Lamb Foundation. It allowed us to make that first knockout animal. It allowed me to get money from the DoD, and now I have money from the NIH to continue to study lamb. but nothing would have happened if I hadn't gone to a Lamposium meeting and I hadn't gotten those initial funds from the Lamb Foundation. And these are funds that are raised by patients like everybody here who gives money to the foundation and contributes to its operations. So I really would be nowhere in this field if it wasn't for the Lamb Foundation. These are just some of the individuals who helped with the work. So that's all I have. I'm happy to answer questions at the ending, but now we're going to move over to our second speaker, and I will stop sharing my screen. Thank you for your attention.
So I am going to go through actually what happens when you donate your organ and it gets sent to us. Let's start from what we knew when we started this work. So we knew that, this is summarizing what Dr. Hammes has already told you, that LAM is a metastatic hormone-responsive disease that predominantly affects women. It causes cystic remodeling of the lung, and that's what gives you the problem. So we really wanted to focus on what's happening in the lung and how it gets to the lung. The Japanese group had told us that almost all patients, if not all, that have pulmonary LAM also have uterine LAM lesions. And what you just heard from Dr. Hammes, he had shown that uterine-specific loss of TSC2 results in LAM-like lesions that are hormone responsive in the uterus and the lung, just like we see in patients. So what did we really wanted to know, do LAM cells in the lung come from the uterus? So the first step to answer this would be really to understand in great detail what these LAM cells are and see if they're the same in the lung and the uterus. Because if they came from the uterus, they should share many of the same features that they had when they were in the uterus. So there is an opportunity with some new technology that we could look at individual cells. So we could look at the cells that were, that we saw in the uterus, and we could look at individual cells that we saw in the lung, and we could interrogate them and find out everything about them. And how we find out about cells is we look at the genes they express. And if they expressed, we can look at all the genes they express. And we also have methods available to determine if the lung and the uterus cells look the same, act the same, and have the same mutations. Because if the mutation is kind of like a birthmark for those cells. And if they have this unique birthmark, if it came from the uterus to the lung, they should have that same birthmark in the lung. So we went ahead and said, okay, these are our opportunities. This is a phenomenal opportunity. But what are the things that have to be in place for us to be able to do that? And so we looked at the challenges. So we needed to get fresh tissue, both from the lung and from the uterus, and we needed it within 24 hours of when it was taken from you, the patients. We know that LAM lung tissue is infrequent, so there aren't very many LAM transplants that are happening, and we wanted to move this research along as fast as possible. And uteri are taken out of LAM patients even less frequently because they don't cause problems. So they're not transplanted and they're not removed unless they have another disease that's affecting the uterus. We knew that the LAM cells are rare. They're mixed with a whole bunch of other cell types, so it's kind of like looking for a needle in a haystack. And they're difficult to isolate and grow. Also, when we look at the tissues, so down here is one of the LAM lungs that one of you patients donated, and this is a piece of the uterus that another patient donated. They really don't look very much the same. And when we look at the tissue, we can't see the LAM cells. It requires us to look under the microscope. So how are we going to find an area in these tissues where we make sure we get those cells? The studies would require a multidisciplinary skilled team that have complementary expertise because there are a lot of steps in the process. The studies are expensive and we need to repeat them in multiple patients. So we need to really understand these cells, we need it from the cells from a lot of different patients. So if you look at the opportunities, they're phenomenal. But if you look at the challenges at the beginning, They looked almost like, how can we do this, right? But one characteristic I think of researchers are their impossible can sometimes become possible. And the Lamb Foundation had such a good infrastructure set up. And if you know, which you do, Frank McCormick, he never takes no for an answer, right? So we can do this. Let's start by putting the stuff we need to put in place. So we're fortunate in Lamb is that we have patients willing to donate their organs. And we knew when the patient is in the operating room, when you're in the operating room, we need families and supporters that are going to be willing to help to make sure everything goes along well. Well, you have a wonderful infrastructure of this already set up. And I had the fortune of becoming involved in setting this up because Sue Sherman and I had talked several times, how can we make this work better? So that was already in place. We had to figure out a way to get the organs from the OR, from the operating room to Cincinnati in less than 24 hours, no matter where that organ was taken out. And again, the Lamb Foundation in partnership with NDRI had a great consent process. They had set up a system that they could alert everybody that the procedure was taking place. They would arrange transport. They got the surgeon and the surgeon's team together with the pathologist and the team at the place where the organ was being taken out so that they could package and send us the tissue in a precise way that we needed to preserve those cells. And that was not easy. It had to be on ice. It had to be in specific media. And then it came to me, and we had to find out some way that we could pick a region from these tissues that was likely to have a high concentration of Lamb cells. So if you think about it, if we want to know everything about you, we would ask everyone who has ever met you to tell us about you, and we'd have a really good picture of you. So that's what we wanted to do with the Lamb cells, right? But that really isn't feasible because we can't talk to everyone. So what we need to do is we need to pick a location where we can talk to as many people as possible that know you. But if we only have the resources to do one location, we have to carefully select that region. Well, from all the research that's been done in Lamb, we knew that in the lung, that these Lamb cells were around the cysts. So I chose to pick the area that had cysts that might have a little bump by them so we could get the most number of Lamb cells as possible. In the uterus, from the Japanese group, we knew that the Lamb cells were more often concentrated on this surface than on this surface. And so we did that. And we devised ways that we could look at this tissue first and find the Lamb cells and then go and learn everything about them. Once we were able to isolate the cells and sequence them, then we had to have someone who could take this huge amount of data and tell us what it means. And then we had to repeat that. So it looked like a very difficult task, but with everybody already on board in the infrastructure, the impossible became possible. This is absolutely amazing to me, that in a rare lung disease, we were able to get 4 Lamb explant lungs, 5 control lungs, 1 Lamb uterus, and 1 control uterus sample flown in from around the country to Cincinnati within 24 hours and get this work done. That is a huge accomplishment that could not have happened without the infrastructure of the Lamb and all of you teaming with us to get this done. We assembled a team here in Cincinnati as well as around the country to help in all the analysis we had to do. And the studies were done on each donor sample as soon as it came in so that we were able to change our procedure if we needed to get more information. So I'm just going to show you a few slides of what this looks like. And it's very easy actually to understand. if we get beyond the language of the research. So from 4 patients that sent us their lamb lungs that were explanted, we were able to look at over 92,000 cells and learn everything about them. Now, I told you the lamb cells are rare and hard to isolate. So in those lungs, out of all those cells, we had 125 lamb cells. But those 125 lamb cells told us everything about them, and they were from 4 different people. So that gives us a good general view of what they are. And from looking at them, we found over 700 signatures of those cells that tell us we're unique from other cells. And so we will be able to identify them now in other locations as well. Okay, so now we know what these lamb cells are, and we know a lot about what they're doing by what their genes they're expressing. So now the next question was, can we now go and see where they came from? Okay, so just like we share some of our origins of our family and our parents and where we grew up, these lamb cells would share, we thought, would share some similarities to the organ in which they grew up in. And so Yan Zhu took all of those, everything she knew from sequencing those cells and said, if I compare them to all the signatures of other cells of other organs in your body, and she compared them to a whole bunch of other organs, what organ are these lamb cells more like? And the uterus came out as a very high, the #1 target by far. So it shared similarities with the uterus, even more so than it shared with other cell types in the lung. So that was good evidence that it might arise there. So what about, just like the lung, we found this small core of lamb cells in the lung that were different from all the other cell types. Can we find those same lamb core cells in the uterus if we take a uterus from a lamb patient? And we were fortunate to have that uterus come to us fresh, give very strong data. And you can see that it has very different cell types, but it shares with the lung this very similar, actually the same lamb cell core. So now we know that the lamb cells in the lung are very much like the lamb cells in the uterus. And these cells have some very similar characteristics to the normal uterus. The next question, now actually, I told you we have over 700 now targets. Like Dr. Hammes was telling you, this offers us all kinds of opportunities to develop more biomarkers, right? And now all the tissue you sent to us has been carefully saved and annotated for multiple different studies, including looking at the lamb lung and the normal lung for these different new targets that we found. So these organs that you donated will be used for years in the future to continue this work. So I can't thank you enough and just highlight how none of this work would have been possible if you would not have come on board with us and been generous in sharing your organs for this research. So the next thing we said, well, do the lamb cells in the lung look like the lamb cells in the uterus? And initially, when you look at the lamb lung and the lamb uterus, they do not look the same, right? You do not see the similar features when you just look at the tissue. But if we look microscopically, this is a cyst in the lamb lung with the lamb cells growing beside it. And this is the lamb lesion in the uterus with a cystic space and the cells growing around it. And you can see those cells look very similar. And there are lymphatics. There are vessels right by these lamb cells where we know the lamb cells can go into those lymphatics and spread, okay? And now we've identified some of the proteins that we think these lamb cells are making to make those lymphatics actually grow, okay? So they're more able to spread throughout the body, which again would be wonderful targets for treatments in the future. And then do they function the same? Yes, they express the same proteins, well, that's in brown here. And these HMB45 positive cells that you hear about used to diagnose the LAM in patients, they are not expressed in every cell, but they are expressed in some of the cells in both the lung and the uterus. So very similar in both the organs. So they look the same, they act the same. So then we said, okay, when they have a TSC2 mutation and it occurs sporadically, that's a very rare event. If that mutation occurred in cells in the uterus and they travel to the lung, both the lung cells and the uterine LAM cells should have the same mutations. And they do. So we give them codes. In the cell, in every cell, you have 2 copies of the TSC2 gene. This patient that donated her uterus to us, we sent the samples to Boston and they helped us sequence, and they found 2 different mutations, one in each of the 2 genes. And we gave them a code. And when they looked at the lung, they found the lung had the exact same 2 mutations. So this is a very rare event. So the fact that both the cells in both locations have this signature, have this birthmark, as we could look at it as, that means they're highly likely from the same source. And if you look at how many cells actually within the tissue that we sent them have this mutation, you can see it's a very low number, 2% to 5%. That's why we really wanted to focus on making sure we got the right tissue that had these cells, because they can be rare. And we were lucky, and we used everything that the research has told us up until now to make sure we picked those right locations so we could find that needle in a haystack. So this is a great story of how everybody working together, we came up with the conclusion that the uterus is a likely source for LAM cells in the lung. And the support of that is the LAM cells, we know them exquisitely now by looking at everything that they're making. And we've looked both in the lung and the uterus, and they look the same, which we call morphologically indistinguishable. They have the same birthmark, which we call TSC mutations in both of their genes. They show close transcriptional identity, which means that everything they're making is the same in both locations. And the LAM cells are retaining some of the features of their birthplace because they look very similar to the normal cells in the uterus. They've just gone off in a different path because they've acquired these mutations. So great discoveries from everybody working together. And the discoveries are going to continue because now we have 777 genes that are unique to these LAM cells and our signature genes from them. So we can look which ones are secreted into the blood, so they can serve as biomarkers for diagnosing, for predicting outcomes, and for testing the therapeutic responses when we test new drugs and you take new drugs to treat your LAM. We have new signaling pathways in addition to the TSC2 or the mTOR pathway. And some of these look like they might make these vessels develop and grow. And so if we could target those, maybe there'd be less spread of the LAM cells. And now we have potential new drivers for LAM progression, and we can explore them as potential targets. So I really want to thank everyone for joining our team. So this, from the study that I just told you today, we've put this together in a publication that's led by Yan Zhu and Frank McCormick. And just for this one study, it took all of these people who are authors on that publication that are shown here, plus the members of their labs, to take your organs that you generously shared with us to find or to support uterus as a likely source in human LAM. So again, thanks to the patients, families, and donors who made this work possible, and the Lamb Foundation with its incredible infrastructure, and the National Heart, Lung, and Blood Institute is also now supporting these studies. So thank you all, and both I and Dr. Hammes are happy to take any questions. So I will stop sharing my screen.
Okay. So can folks hear me? This is Jessica again. Little thumbs up.
Yes.
Yes.
Great. Thank you. I really want to thank both Dr. Hammes and Dr. Wickenhauser-Brokamp for their presentations. Usually discussions of cells and mice and research, I glaze over. And so I found this very accessible, very useful and helpful, and I really, really want to thank them both. And I also want to thank the Lamb Foundation. We hear how important it is to research, and we recognize, you know, how important it is for us to be able to come together in sessions like this through the work of the Lamb Foundation and through the work of all of us, and doing advocacy and promoting and donating and doing things to support this foundation that in turn really supports us. So again, thank you so much for this evening's talks. And there we go. I'm trying to get my screen set. So we do have a few questions that came in earlier through email. So I'll start with those. And then I've also been keeping an eye on the chat. So we have quite a few questions to cover. So the first from Celia Benavente, a lot of background, but the questions really are, she's 54 years old, supposedly in menopause, but has had intermittent bleeding, which now seems to resemble a regular period. So questions about estrogen levels. Also, there's other, she also has a few other medical conditions, Hashimoto disease and hypothyroidism. So questions are, is there any connection among these various diseases? What are estrogen levels at play? Maybe you can just talk a little bit more about that. We heard quite a bit about the uterus and estrogen. So how does this all play together?
Okay, I can try and tackle it. And I've been looking at the chat room, so I'm going to try and do like 10 questions in one answer. So I think there's a lot of people asking questions about two things. One, the difference between an ovary and a uterus, the difference between a hysterectomy and an oophorectomy, and how estrogen plays into it, and how the uterus plays into it, and also how somebody can have their uterus removed 10 years previous and then get diagnosed with LAM. And those are all excellent questions, and I'll attempt to give my at least my thoughts about it. So as far as a specific question, you know, menopause, it can vary. It can start at, you know, some people go through an early menopause in their 30s. On average, people go through menopause when they're in their 50s, but sometimes it can be even later. It can be 54, 56. It's really, you know, it's a bell-shaped curve, and it's very, very broad. And the description of feeling like you're in menopause and being sure you are, and then a year later you get another period is not an uncommon thing. I don't think it has anything to do with LAM. It just has to do with, you know, with the inconsistency and when people's ovaries finally stop working and when they go into menopause. And so I don't think there's a relationship, but the caveat I'll make, and I don't know the answer to this, if you're in the chat room and you can write it, let me know, are you on sirolimus or another mTOR inhibitor? Because mTOR inhibitors do have effects on ovarian growth and follicle growth, and they also have effects in the uterus and can cause uterine bleeding. And so, you know, all bets become a little bit off when you're on sirolimus. And so if you are, that could also be contributing to the process. But if you're having a period, that means your ovaries are working, and that means your uterus is responding to the ovary. And so it does, you know, so estrogens are being made and could have an effect on LAM, you know. And again, when you go through menopause and estrogen levels finally go significantly down, it does appear most patients do seem to have a calming influence. So the ovaries make estrogen, and then estrogen stimulates the uterus to grow, primarily the endometrium, which is where the blood is coming from. So so the answer is so what I would say is when you have an oophorectomy, you remove the ovaries, but most people get hysterectomies, which just removes the uterus, and the ovaries are still present. So it's a common question I ask women: have you gone through menopause? And they say, my uterus was removed. And I say, that's true, but that doesn't mean you've gone through menopause yet. You get hot flashes, you get all the symptoms of having low estrogen when you actually go through menopause. So they are two different surgeries, but estrogen is seemingly what stimulates the uterus to grow. So could you, if you have a hysterectomy, can you get LAM 10 years later? And does that disprove that the uterus is the origin of the LAM cell, which a lot of people were asking? And I'll be interested to hear what Catherine had to say. My own feeling is this: LAM is a very slow growing disease, and it's very possible that you could have had a metastasis many, 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 many years prior that was just growing very, very slowly and eventually led to disease. But the other caveat I'll make from everything we just said is it may not be that in everybody LAM is actually metastatic from the uterus. But what is clear is that those LAM lesions in the uterus or in the lung look exactly like the LAM lesions in the uterus. So even if one didn't come from the other, they share the same program and they share the same mutations and they look the same. And it does suggest that there is a similarity there that we can take advantage of. So doing the expression studies like she was talking about, I think are really going to help us understand things. So it may be that there's just a program in some patients who have LAM lesions, and it makes it look just like a uterus. And again, it doesn't necessarily have to always be cause and effect. It could just be they're incredibly similar. But either way, it's just useful information that can help us. So I'll stop there.
I can just touch on a few of the questions. Can you hear me?
Yes.
Okay, I just touch on a few questions in expanding on what Dr. Hammes said. So one thing is leiomyomas or fibroids in the uterus are not LAM lesions. Okay, many people have those. Those are not related to LAM. It's just that's what the uterus is typically resected for. And then we can look at it to see if there are any LAM cells there. Another related question is somebody asked that they have had their uterus removed and there was no mention made that there were LAM cells there. That is, the LAM cells can be very, very sparse in the uterus, and you can't see them unless you examine the uterus under the microscope. So we don't examine the whole uterus under the microscope. That's what the Japanese group did, which was phenomenal. And the vast majority of women with LAM have only very focal LAM cells in their uterus. So it is not surprising that if you had the uterus removed that they didn't find LAM cells, because we do not routinely extensively sample the whole uterus. And the uterus, the LAM cells in the uterus don't really cause a problem or give you a symptom that you notice. So although that's where they may start, that isn't your problem clinically or by symptoms. So I just wanted to make all of those points clear. The other point was if in some patients or in some LAM patients, the LAM cells may come from the uterus, but that doesn't mean that in other patients, the LAM cells may come from their kidney tumors, their angiomyolipomas. And we are looking at some angiomyolipomas, the kidney tumors, by this same analysis to see how closely they relate to the LAM cells in the lung and the uterus. So there is a possibility that there could be more than one source. But
I guess the only caveat I would say with that is that males get AMLs fairly frequently as well. So they seem, I would argue they're probably a little less estrogen sensitive or significantly less estrogen sensitive. So it does suggest maybe, it'll be interesting to see what they find, but I do think they're a little bit different. And in fact, in the like 621-101 cells, which came from an AML, estrogen receptor levels in our hands are super low, and they're not really estrogen sensitive as opposed to, true lamb cells. So it'll be interesting to see. But I echo the point. The number one reason to get a hysterectomy is a leiomyoma or a fibroid, because they cause pain and bleeding. But these are big, and you don't need a microscope to see them. And as Dr. Wickenheiser-Brokamp mentioned, the lamb tumors you're right, it's Herculean, because you have to look at this giant uterus to find a 2 millimeter lamb lesion. And so if you're not thinking lamb, you're not going to find it. But if you are thinking lamb, you will almost always find it.
Great, thank you. There's so many connections and questions about these different connections. A few things came up about specific, you mentioned stain positive HMB45. So there was a question early on about what if a tumor is not stain positive, and then also a question about the GPNMB, and if there are blood tests available to check those levels. So any thoughts on that?
Well, for part one, I'm going to totally defer to the pathologist.
I'm happy to take that one. So we have known for a long time that not all lamb cells express HMB45. So it's a very specific marker. If you find cells in the lung that express HMB45 or are positive for HMB45, it's very specific to lamb. But you can have lamb and not see HMB45 positive cells because not all lamb cells express it. So that is another reason why we need to find some more markers of lamb cells, because it can be easily missed in a biopsy if the pathologist just says it's not lamb because there are no HMB45 positive cells. It can still be lamb without those. And I think we have found that actually in some patients, only the minority of their cells express HMB45. So that does not eliminate a diagnosis, and it tells us that we need to find even more sensitive diagnostic markers. And then I'll defer the other question to Dr. Hammes, because that's a discovery and that's a potential marker that we could use.
But I think the key word is potential. I will say that, as you saw, only a couple percentage of those lung tumors have lamb cells. And, if you're on the lower end of that curve, you may just miss it, and the expression may be there. It's just too low to detect. And it may also depend on what part of the cycle you're in and where your estrogens are and how old you are and whether you have ovaries and whether through menopause. There's so many things that could be affecting whether that stain comes back positive that it wouldn't surprise me. GPNMB is really interesting, and we're interested in it, but it is still too early, because I can tell you we've only looked at 10 patient samples and 10 controls, and the data I showed you are really encouraging, and we have another 30 of each. And we were about to do it in mid-March, and then everything got shut down. So it is on our list now that the lab is back and up and running. It's on our list to do far more patients, because we really need to do a lot of patients. And then we need to be able to compare it to VEGF, which is another one people use, marker people think will be useful, or compare it to their FEV1 to see whether there's any correlation between the levels of GPNMB and their lung function. So there's a lot of work that has to be done. So everything I'm showing you is preliminary. That work I just showed you in the blood plasma is not published. That is fairly recent and just something interesting and exciting. So I couldn't resist but talk about it.
Thank you. We appreciate that. A question about what's known between shared molecular and signaling pathways between ER positive breast cancer and LAM.
That's one of my favorite subjects, and that's something we're very interested in studying. So we're doing a lot of work looking at what estrogen does in lamb cells versus breast cancer. And I think that there are common pathways that appear to be activated. There are both transcriptional signals by estrogen, and there are also non-transcriptional or non-genomic or more signals that don't involve changes in expression of RNA and of proteins that are also promote growth. And it's always been interesting to me that in humans and in mouse models and in cells, there seems to be this connection, this estrogen connection, yet those studies have not been done in humans where you really do estrogen deprivation studies using aromatase inhibitors, just like we do for breast cancer. And it's harder to do because there's so far fewer LAM patients than there are breast cancer patients. But there are these connections that I think, to me, if there's a clinical trial that I would love to see, it would be and we know exactly how to do it because we do it all the time with breast cancer. So it's not like you're developing new protocols, they already exist. And so it'd be a very interesting study to do because if it had a profound effect, we'd all really be excited. And if it has no effect, then I'll stop studying LAM signaling and or estrogen signaling in LAM tumors, and I'll move on to something else. I just need to know biologically, does it do anything in humans? If it doesn't, then we need to think of something else. But I suspect that it probably will. So there are these connections, but there's still a long way to go.
And your the data that we got from the organs that you've donated, they will help in answering those questions because now we know at a single cell level which cells we can say, okay, if they're expressing estrogen, what else are they expressing? And the other cells that are there, the ones that aren't expressing hormones, how are they different? And so that will help us target different, know which pathways we need to target and which cells we will be targeting. The other thing is I'll tell you that some people who donated their tissues have been on sirolimus, and it looks like there are definitely differences in the cells, the cell identities and what they're doing when they've seen sirolimus before. So we're very interested in determining what's the difference in ones that were treated versus not treated.
Thank you. And I think if we could, we're at 8:16. If we could keep you for just a minute more, because there were some questions about sirolimus and everolimus affecting estrogen levels, as well as progesterone affecting estrogen levels. So any additional thoughts on how those interact with estrogen levels?
Sure. So a couple things. Number one is it was interesting in our model where I showed you get rid of estrogen and tumors don't grow. Well, when you look at mTOR activity in the absence of estrogen, it is also quite low. So even though you've gotten rid of TSC2 and you think mTOR activity would be super high, without estrogen, it isn't. Again, suggesting that you need an accelerator. I always think of it as a car on a hill or a car that's on a flat road. If you take your foot off the brake, the car is not going to move until you step on the gas. And so in a cancer, if you get rid of an inhibitor, it's still not going to grow unless something else is stimulating it to grow. And so I think those studies with RNA-seq are really going to be critical in comparing sirolimus to non and postmenopausal to pre. I think it's going to be really cool. So estrogen and progesterone. So estrogen is made in the ovaries and it goes cyclical. So normally estrogen levels in men and women are about the same. And it's only during ovulation in women do estrogen levels go up to, 5 or 6 times that of men. And it stays that way for about a week, and then it goes back down. And what happens is when you ovulate, along with your oocyte comes something called the corpus luteum. And the corpus luteum is what makes progesterone. And then progesterone, if the egg becomes fertilized, the corpus luteum continues to turn into a placenta, and it continues to grow and you continue to make progesterone. But if you don't fertilize the egg, then eventually the corpus luteum dies and progesterone goes down. Progesterone is a really potent inhibitor of estrogen production in the ovary. And so it's really important in a normal cycle for a woman, because when estrogen goes up, it kind of shuts down, or when progesterone goes up at the ending, it shuts down estrogen production and it lets the whole cycle start over again. So we give women progesterone in order to suppress their ovaries, and that actually is a great form of birth control. There are some problems with it, because if you suppress estrogen too much, it might have ill effects on the bones and other things. So I don't like giving progesterone, but 20, 30 years ago, it was very common to give progesterone. And the thought was in LAM, if you gave progesterone, you would turn off the ovaries, estrogen levels would go down, and that might turn out to be helpful. The problem is that it doesn't knock estrogen levels down to 0. It just stops you from making estrogen in the ovary, but your adrenal gland still makes precursors to estrogen, which can then be converted to estrogen. So men don't make any estrogen in their testes, but they make precursors in their adrenal glands that are then converted to estrogen in various regions of the body. And so just giving progesterone, or even just removing the ovaries in a human, is not really enough to get rid of all your estrogen. And that's why in breast cancer, they also give aromatase inhibitors or tamoxifen to try and really block either estrogen production or estrogen signaling. The problem with tamoxifen is in the uterus, tamoxifen actually stimulates growth. It doesn't inhibit growth. In breast, tamoxifen inhibits growth. In the uterus, tamoxifen stimulates growth. And so there are some studies in the 70s where they gave individual patients tamoxifen and they got worse. And it again argues for a more uterine-like phenotype that actually gets stimulated by tamoxifen. Jessica, your cat just got
Yes,
right in
my
front
cat
of you.
is stealing the show. I'm sorry.
Great.
You need some attention right now. I apologize. So there were a couple of questions about surrogacy. One more related to estrogen production during IVF and letrozole, and the other was more related to stopping sirolimus use before egg retrieval. So can you speak on either or both of those issues?
All these endocrine questions. So see, the token endocrinologist, that's what happens. So as I mentioned, sirolimus does have effects in the ovary, and there's a couple papers published primarily looking in kidney transplant patients who would take sirolimus as an immunosuppressant. And like almost 50% of them develop abnormal uterine bleeding, and a huge percentage of them will develop ovarian cysts. See, I can't even get my terms right. They will develop ovarian cysts. And so there are a lot of LAM patients who do develop ovarian cysts because of the sirolimus. And so you do have to keep an eye on that. They're benign, but they can grow and they can be painful sometimes. So certainly if you're trying to get pregnant, I think that everybody would tell you to stop sirolimus. And if you are pregnant, I think most people are unsure what sirolimus would do during pregnancy, but are pretty sure it would do good things. And so I think you really need to stop it when you're trying to retrieve oocytes and also during pregnancy, which is unfortunate. The retrieval process, if you need to do oocyte retrieval, you do have to stimulate the ovaries, and it does make estrogen levels go up. And there have been examples where patients have had pretty significant worsening of their LAM when during these retrieval processes. It's tricky, but not everybody, but many people. And so there are some low estrogen procedures now that they use because they do this in breast cancer patients all the time. And so they've got tricks, but you really need to go to somebody who knows what they're doing, I would say, and who can tell you all the benefits and risks. And it is something you have to think about very carefully, I think, because the difference between breast cancer and LAM is once your lungs get worse, they may not get better. And breast cancer, even if they do have 3 or 4 days of estrogen that they need to get, that might affect growth, it's pretty minimal. And so you can work around it, but it's a much harder, once you lose lung function, it's lost. And so I think it's a lot more, it involves a much deeper conversation. You really have to be really sure it's what you want to do.
I wanted to answer one question that's in the chat box too, because I think it might relate to a lot of different LAM patients. The question was, is there current research applicable to both sporadic LAM and TSC LAM? Absolutely. The lung-uterus connection is shared in both sporadic and TSC LAM. The studies we did were on sporadic, but we know from the studies the Japanese group did is that whether you have TSC LAM or sporadic LAM, they found LAM lesions in both in the uterus. So we think it will apply to both.
Thank you. And I'm going to take a little moderator privilege and ask a question that pertains to my situation more specifically, which is about the question right above sporadic and TSC LAM, about lymphatic masses causing things like chylous effusions, which is something that I experienced, and what kind of research is being done in that area.
So I can address that initially. We're really focusing on understanding the LAM cell better. So it won't matter if we can target the LAM cell. Our thought is that if we target something that's expressed in the LAM cell, regardless of where it is, it should be effective. So our hope is that whether that LAM cell is in the lung, in the uterus, in the lymph nodes, in the kidney, in the lymphatics, like in your case, that it will affect those cells as well. The second thing that I think particularly is exciting and pertains exactly to that is if it looks like the LAM cells are expressing some things that make the lymphatics grow, which would make the lymphatics more accessible to them. So if we could stop that, it might help the LAM disease in the lymphatics, which I think is an exciting new opportunity.
So another thing that I didn't talk about at all that we've become very interested in recently is how the immune, and many people are interested in this in many tumors, is how the immune system, we talk about lymphatics and we talk about the immune system and how the immune system interacts with cancers, and in this case with LAM. And you know, we always think that an immune response is a good thing, but what we are seeing right now, for example, with COVID, is that a robust immune response sometimes is a bad thing because it causes release of cytokines and other things that can actually have so much off-target effects that it actually turns out to be a bad thing. And it's the same with cancer. Inflammation is, which we think is a good thing, it helps us fight infection, but in cancers, inflammation actually can stimulate cancers to grow. Cancers are really smart and really good at hijacking our immune system and trying to use it to their benefit. And they're also really good at hiding themselves from the immune system so that the cells that are supposed to kill it can't kill it, and the other inflammatory cells are actually stimulating it to grow. And so there are a number of people who have been funded through the LAM Foundation and who are part of this organization and on the same board that Catherine and I are on, and our lab is also interested in how we can manipulate inflammation in the immune system and really try to sometimes you might not have to directly attract the LAM cell, you can do it indirectly. And using that same technique, all the information about the immune cells is in the data that was just shown to you. There's been a focus on the LAM cells, but you can look at any kind of immune cell and we can share that data and use it to try and, you know, benefit our studies on the immune system. So it's a powerful, powerful amount of data that's been generated since then.
So not only did we figure out the identity and exquisite detail of the LAM cells, those 125 LAM cells in the lung, but those 90,000 cells around the LAM cells, we can now compare those, how they're different from the cells in the normal lung. So the inflammatory cells, the cells that line the cysts, the vessels, the lymphatics, and all of those interactions now can be interrogated. So it's exciting because there's enough there to keep a lot of people busy for a long time.
Thank you so much. We are going to wrap this up. I know there's still a few unanswered questions in the chat. I'd ask the doctors if you saw anything in the chat that hasn't been answered yet, or if you think there's anything more that you would like to just reinforce or share with us before we let you go.
The one thing that I saw a couple people ask is pituitary and how that relates to LAM. And as far as I know, there's really no connection. The pituitary is located, you know, just outside of your brain, and it basically regulates a lot of endocrine responses. So the pituitary does control estrogen production in the ovary, but it's all, but as far as if you have a pituitary tumor, is it any relation to LAM? The answer is most likely not. You know, in TSC, you get lots of different types of tumors. You get skin tumors, you get tumors in neurons or in your, in the neurologic system, you can get lots of different, or you can get the renal tumors as well, and then the LAM tumors. And so there are some cells that seem to grow when you lose TSC, but I don't think it's ever been reported in the pituitary that I know of. I don't know of any relationship. So I think if you have a pituitary tumor or adenoma, it's probably unrelated. 10% of people do have pituitary adenoma. So that means 1 in 10 of you probably does if we look.
And I just want to end by thanking everybody. And somebody asked if they said we have your lungs, so thank you. We have saved every piece that everybody has given us. And I don't know that I can get you a tummy tuck if you send your uterus.
That was the best line in the chat room. Thank you for that.
Was a good one. But I just want to thank you again. And it is amazing. You know, often the patients tell me they don't understand the science. It's like a foreign language, but your questions that you ask really show that you guys have a very good understanding of the disease. And I tell you, it's just wonderful to us to be so engaged with you, and you are all so motivating. So thank you for all you contribute and all you do. And it really is a team effort. And I'm so happy to be on the team. It's really, like Dr. Hammes said at the beginning, this is really a group like no other that I've ever worked with. It is just, it's fun. And there's not very many rare diseases that cause the problems for people that this disease causes, where you can actually enjoy being together and talking about it and finding out more. So thanks for your help. Thanks for your being engaged in the research, and thanks for everything you've done.
Yeah, thank you so much to both of you. And, you know, I know we had to make adjustments from being in person at Lambposium, but this has been such a great opportunity to get time with you to really be able to ask these questions and to get all the information that we would have gotten if we were in person in Cincinnati. So we really, really appreciate that. And I'm going to wrap it up. I think I'll hand it over to Shar for a minute.
Thank you, Jessica. And doctors, again, thank you both so much for being with us. I think Jessica said it all pretty well, but the appreciation runs deep. I know from myself, from the foundation, from this entire crew that's on here with you tonight. So thank you very much. I can make no better case than Dr. Hammes and Dr. Wickenheiser-Brokamp have made today than for you to register with NDRI if you are looking to have surgery, or if you're even contemplating tissue or transplantation and would like to donate your tissue. You can reach out to the foundation. You can reach out to me through Circle of Hope, and we'll get you set up so that we can continue to provide them that very precious tissue that they need to continue doing the amazing work that they're doing. So thank you very much. And I do believe our CEO and fearless leader, Sue Sherman, is on with us tonight, and wanted to make just a couple of closing remarks before we move into the group discussion. So Julie, if we can unmute Sue.
I think I'm unmuted. Can you hear me?
Excellent. Yes, thank you.
Wonderful. Well, I'll only take a moment to express my gratitude as well to our speakers, Catherine Wickenheiser-Brokamp and Steve Hammes. Your enthusiasm and commitment to this community comes through in your presentation. So thank you for all that you do and for your evening and the time that you spend with us. A shout out to Charlene Dunn for her leadership in the Lambposium in Your Living Room series. We're looking forward to many more of these, and with 100 to 200 people per session, this is, though not the same as Lambposium, a remarkable recovery and a way to share information. I also want to thank Jessica for moderating, and for Julie on our staff for hosting the technology, and Lisa Catalano for generously sharing her story on the patient focus. From here, I just want everyone to know that we are focused on the future. We are mission driven, which is to find better treatments and a cure. We are grateful for your time, everyone on this call, and your financial generosity. I can't think of a greater example of being such a tangible example of how we raise money and how we spend it and how we move the science forward and how you have access to it. So thank you again. Be resilient, stay hopeful, and happy Worldwide Lamb Awareness Month. Tell everyone you know about this journey because we're going to get to the end of it. Thanks to all of you. Thanks, Shar.